Okay. So good morning, everybody. Thank you for joining us again in our grand rounds. And it's a real pleasure for me to introduce Paul. Paul's become over the years a really good friend. I have this long list here of titles of Paul, which is very long. I'll give you some of them, but he's actually not a cardiologist. He's actually a biomedical engineer. He's one of the few biomedical engineers who's not just a professor of biomedical engineering, but he's also a professor in cardiology. He's a professor in integrated biology and physiology and at the Carlson School of Management at the University of Minnesota. He serves on multiple boards, and his main research areas has really been translational systems physiology. He's trained over 200 graduate students, postdoctoral fellows, and medical students, authored almost 300 articles, 100 book chapters, 5 books. But maybe I'll share something personal, how I came to know Paul. So I met Paul about 10 years ago when I traveled to his lab at the University of Minnesota from when I was still living in Italy. And at that stage, I was publishing lots on coronary bifurcations and stenting and thought I really understood the field really well and was a sort of thought leader in the field. And then I went to Paul's lab. And for the first time, I got to see what I actually did in patients. I got to see inside an artery and how I was manipulating stents and putting stents into a bifurcation. And that experience of spending a day in Paul's lab impacted my clinical practice. And I've always thought about that, how someone who's a biomedical engineer, who I didn't know about, was doing this work somewhere else that really affected what I do every day. And since then, I've gone to Paul's lab multiple times. I've taken some of our faculty, Andrea, Manaf, Asma. We've all gone to work in Paul's lab to teach, to understand better how our devices interact with cardiac anatomy. And Paul has really created a unique way that nobody else in the world has been able to do and recreate of how to visualize anatomy in a heart. So with the fellows, I would suggest you go online. I sent you the link and see his library of anatomy. For me, whenever I want to understand how the mitral valve works, the tricuspid valve, I usually go look at one of Paul's videos. But it's not only impacted interventional cardiology. If you're an EP, Luigi, a lot of what you do now with leadless pacemakers, with ablation and mapping, comes from Paul's lab because Paul works really closely with Medtronic. And a lot of the work Medtronic does is done in Paul's lab. But he's also done stuff on organ preservation and organ transportation, and maybe he'll talk about that. For the fellows, take one of the cards here of the Visible Heart Lab. There's a QR code in the back, and you can download some of the apps that Paul's team have created. I have them on my phone. When I'm trying to understand TEE and different positions of a TEE probe, he has all these simulators that you can have on your phone, and you can use them to really understand cardiac anatomy. They're really incredible. But Paul, thank you for coming. I haven't really talked about your topic today because I learned about the black bears from you, and I think this topic is really an area of great interest that I'm going to leave for you to tell our faculty about. But once again, thank you for taking the time. It's a real pleasure to have you.
Thank you for the invite, and Dr. Garcia as well. It's been a pleasure to be here, and it's always fun to talk about the translational aspect of my work. I'm actually a human physiologist by training and an anatomist, but I do teach in biomedical engineering, and most of the grad students in my lab are biomedical engineers. But I have one of the coolest jobs on the planet because not only do I get to do the reanimated hearts all the time, I actually do field work a couple of weeks out of the year in the middle of nowhere studying hibernating black bears. So I'm going to kind of try to combine both of those today. I'm going to talk to you about the translational research I do and how that impacts what we do in the lab and vice versa, go back and forth. Talk about some hibernation physiology. Bears are amazing animals and amazing hibernators, and I'll discuss some of that. And they upregulate a whole bunch of hormones and burn fat during the winter, and then we're trying to make that translational back to human clinical medicine. And then, study this pre and post conditioning and how it could impact not only what we do in the lab, hopefully clinical medicine. So we do a lot of different things in our lab. It's a large lab, but basically we do things where we do target drug delivery. We do preconditioning agents in the pericardial space. And that is done routinely in the lab. We typically will do that for every prep an hour before doing omega-3 fatty acids or small lipid. That's based on the bear work, and I'll talk about that. We do muscle force studies, and I did muscle force studies in hibernating black bears as well as patients in intensive care units. Doing a lot of work on transplantation. University of Minnesota has a rich history in transplantation medicine and research. Huge outreach mission of the lab. We do isolated tissue bath work, biomechanics, but still the centerpiece of the lab has been the Visible Heart Project. And we've collaborated with Medtronic on that for the last 27 plus years. All the different groups from, valves, stents, ablations, pacing systems. And see, we are a preclinical research lab, but we're early. We're not a GLP approved lab, good laboratory practice. So we get the early stuff, I have a cool job. They bring me a novel concept or technology, and I have to try to figure out how to do a study on it. And to me, that's the fun part, right? And it takes a lot of creativity and intuition. So the joke of the lab is I probably put in more cardiac devices than anybody in the world, but I've probably broken more cardiac devices in the heart than anybody in the world. Because the early concepts, and we'll try to glue them back together or weld them back together in the lab. But it's really a team sport, right? So I have a huge lab. We have a number of staff here. Currently, there's 8 PhD students, 3 master's students, a couple postdocs. We have over 15 undergrad and volunteers, which make us one of the larger labs on campus. And to me, this is really the most important part of my job. The coolest part of my job is training individuals and watching them, launch their careers and where they're going. And it's really them that keep they're the brain trust and keep me still at the U and still active, I guess. So the Visible Heart Project really started in '96, and we got funding from Medtronic. And basically, we were doing isolated guinea pig hearts, and then a group from Medtronic, and it was my brother-in-law and Tim Lasky, said, hey, that'd be cool, but if you could do that with a large mammalian heart, we could pace, that'd be even better. So we said, why not? And we started doing it. And, it's just using a cardioplegic buffer to stop the heart, or attenuate all the great vessels. And then you can do a Langendorf perfusion mode or a whole 4-chamber working mode, full functional mode of the heart. So, we've kind of published that and been doing that continuously. And then this was first described by Oscar Langendorf, who was a German physiologist from Rostock in the late 1800s. So it's nothing new, except we have all the cool technology and videos and equipment to be able to see all this. And so this is just showing a video clip from many, many years ago where basically we can have a Langendorf mode, which means the aortic valve is closed. We can push a button and it can go into a 4-chamber functional mode. And we're a little bit like Hollywood, so we'll line up all our cameras to get the views that we want. And so, again, a little bit more details on the procedure. This is how we remove the hearts. I've watched Indiana Jones, this movie, many, many times. Carefully studied this. This is the quickest way to remove a heart for reanimation. And you just, go in there. And I have super strong hands, so don't make me angry. Because, I know this technique. I know that's not true. I don't do that method. But I do wear the headdress every time. And then I thought, this is such an old joke. I've been saying this for 20 years. But then Indiana Jones 5 just came out. So I feel I'm younger than Harrison Ford, so it's okay for me to show this video. So, we reanimate animal hearts on a typical basis, large mammalian hearts. And typically it's porcine hearts, pig heart. And, a pig will live for 5 or 6 years. you can pick the size heart from the size animal you want. And typically we're taking animals that are 7 or to 9 months old, and their hearts are 450 to 500 grams. And so they're lung adolescents. We do ovine, mini pigs. We've done some canine hearts. There's a canine heart with a chronically implanted encapsulated lead in it. This is the Philips Spectranetics laser we had in the lab, and we're just showing how that laser will cut through fibrotic growth. So that's a cool part of this, is that you could create all these videos showing technologies and educate people on them as well. The lab is set up. You know, we have state-of-the-art equipment. We're actually just ordered a new Philips fluoral system. We got a brand new 40 echo system, but we have endoscopes that you can put inside, 2.4 millimeter scopes that will fit down the coronaries. We have OCT as well. And so basically, when we're trying to get these images, you're like Hollywood, right? So you're going to line up all your imaging, multimodal imaging, and then you're going to start your procedure and you're going to go. And so this is, you know, kind of the fun part of the lab. And again, it's a biohazard area because we've also had the privilege to reanimate human hearts. So we've actually done 98 hearts since 2000. The last one was a month ago. So these are hearts from organ donors where the hearts are not deemed viable for transplant. They're recovering other organs. And just like a organ transplant, we got to get to lab within 4 to 6 hours. And it had to have a reasonable ejection fraction, and it has not to be cut up. So the liver team couldn't have been aggressive and taken the whole right atrium. They can't do a lung transplant. There's no left or right atrium, and they can't look for valve harvesting. So it's rare. Plus, then I have to have that team of students available in the lab because I can't do this myself. So it's a rare event that you're going to do this. But when we do it, you know, the hearts are cardiopleged. We'll flush that out. Usually the atria will start contracting, and then you shock it, and most of them will go in a native sinus rhythm. And they'll stay viable for some 5 to 7 hours. The first 2 hours are dedicated to collect all the functional images that you'll see on the Atlas website, because we feel that with these gifted organs, we have to gift all that knowledge back to everybody, and that's been a mission of the lab. And then after that, the grad students that are doing work on isolated swine hearts can do it on a human heart. They can then work translational. And then after that, Medtronic will bring in various devices. Because it's a human heart, the concept is that we have to be able to publish or present that publicly in a reasonable time period. So this is the arrangement we've made over the years, and you'll see some of those images we've shown here as we go. And we've also done, you know, heart-lung blocks, and basically, so you basically have a visible lung in a way, but it's fun to pull back in these pulmonary veins. And you can actually see the native pulmonary vein ostia in the left atrium. Again, described by Newell Martin in the 1880s. The difference is we're doing this without blood. And we have all these great oxygenators and everything else, and then the cameras to go inside. And so you can see me pulling the scope back out of the lungs. Now I'm coming back into that left atrium, and I can see all that anatomy. So we've done, you know, the cryo balloon ablations there, being able to watch it seat in there, and it's, you know, collecting all these educational images. And then you can go into the pulmonary arteries and pull back through that retrograde, and you can see all this beautiful branching of all these vessels. And it gives you just a totally different perspective as a human anatomist or as a clinician, of understanding how complex these tissues and organs really are. And then as you pull back here, you're going to see the pulmonic valve. And, you know, this is where we've done implants of the Melody valve. And a couple of weeks ago, we did a Harmony valve in a swine heart. And this just actually shows what you can do is you can have a view of the valve from above and below, echo and fluoro simultaneously in there. And typically we try to do the multimodal imaging and all these specimens as we go. And this is from 98. And so this was a Medtronic 5076 lead. Dr. Tim Lasky and I were working on it. We collected these images. He brought it to the FDA for edification FDA panels to get FDA approval. So we hope it expedited it, but I think they put in over 3 million of these leads now worldwide. And again, it's kind of fun to be able to be part of those kind of things as well. So here's Goran Stankovic. He's visited the lab, the same thing as Zim would have done. We can have function on there. We can have the cameras inside the coronaries. Here's both wires in. We can watch the balloon inflate the stent. We can have the fluoro. We can run OCTs. You can be heavy on the fluoro pedal because basically it's an isolated heart. And so you can do as much contrast as you want for educational video collection. And we've done live webcasts. So the live one we did was to Euro PCR a couple of weeks ago. Dr. Stankovic was in Paris. We had Dr. Rana Davies and Jorg Reifert in the lab, and they were our operators. And we did live case bifurcation sent to Paris to be able to visualize that. So again, when we're doing all that, we're doing Zoom and everything else, but the heart better work, right? So you got a live audience like here. And so we actually try to figure out how we're going to get these hearts to always work well. And so we've done a lot of preconditioning studies of these hearts. Then the other things we've done, the latest thing is Michael Bilecki, one of the PhD students, we're looking at, all the commissure alignments of the aortic valves. We're doing stent and stent. We're actually doing chimney and snorkel procedures. And then what we have in our lab is we have our own micro CT. So we can take these hearts afterwards, micro CT, and get down to a 15 micron resolution of device tissue interface. And then we can do 3D computational models and 3D printing. So we come to the lab, and I've given, I think, Azim some of these 3D prints as well. And now we have actually better materials so you don't break them. They're not as fragile, so I'll have to get you some new ones. But again, this is the partnership is that the grad students are doing areas of research that are anatomic and kind of state of the art in the field at the same time as we're partnering with Medtronic. So human heart comes in, the goal is to get all the functional anatomy, get 3D echoes, do fluoro, do pressure volume loops, do electrical mapping. We have mapping catheters. We actually have the cardio insight system, the Affera system in the lab that we can actually reanimate the hearts in. And we'll fix the hearts. We'll make the computational models. We can do diffuser tensor MRI, get fiber orientation. And then we're doing, with a collaboration with Helena Dobrzycki's group in Manchester, the whole conduction system of the heart. So now we're starting to create a library where we have all that from reanimated hearts. And so then we can really understand if you're putting in an aortic valve, where that left bundle branch is, or if you're doing left bundle branch pacing, what's the orientations of that relative to the septum. And so these are some of the latest things that we're doing in the lab. And we've done this with other organs. So we have a visible kidney, we have reanimated lungs. This is human lungs that we're reanimating in the lab. These on the organ care systems out of Transmedics out of Boston. And so we've been able to do a lot of research on that. As you know, this system is $500,000. Each cartridge is single use. So we've made our own more research test bed for $2,000 to $4,000, we can do a reanimated lung study in the lab and start to look at post-conditioning agents to how to better take these organs and preserve them or post-condition them. Again, most organs that you're recovering for transplantation are marginalized because they're coming out of a donor that's sick and marginalized, and it's a hormonal mess, and it's all the different catecholamines to keep perfusions and pressures. So then when you take those organs away, flush it out, you're actually, you can post condition them. So you can take a marginal organ and say, yes, this is actually a good organ for transplantation. So that's been a focus of the research in the lab. And, for a long time, I was doing muscle force studies. We still have collaborations going on that. Hans Gintz in Basel, Switzerland. We just actually published a paper a couple of weeks ago where we actually monitored patients with neuromuscular disorders in the ICU for 5 years, and tracking, their when they have recurrent visits to the ICU, what their muscle forces are. And so I've been doing that work as well. And then all of a sudden I get a call out of the blue saying, hey, I heard you're studying muscle force in patients in the ICU. How would you like to study a population of individuals that don't get weak, even though they're immobilized for 4 to 6 months? And the call was from Hank Harlow, and who's a zoologist, biologist at University of Wyoming. I said, that would be awesome. He goes, well, I'm studying hibernating black bears. And I'm like, that'd be awesome. So I'm from Minnesota. I love the outdoors. And then over the years, the collaboration has continued and grown, and we've had a whole cast of characters that have participated in the black bear research. But again, what's kind of interesting to put this in perspective, something on the order of 70 to 80% of all healthcare dollars in the US are spent in the last 6 months of a patient's life. A lot of that is spent in the ICU. A lot of that is spent on patients on ventilators. And there's really no good way to assess them or improve their function or wean them off a ventilator. And so we're thinking that maybe the work we're doing on hibernating black bears is a way to do that. So we actually bring force devices in the ICU, so we can actually track their muscle wasting or loss or regain of these patients in the ICU. And just for perspective, this is a study we published, and it was an N of 7 because about 50% of the patients died while we're trying to do the monitoring. But basically, they'll lose anywhere from 25 to 75% of their muscle force in a 6-week period of time, and it takes them 13 plus weeks to recover that, to be able to wean them off the ventilator. Huge clinical problem. So again, phone call from Hank, 1997, same year we're starting the Visible Heart Project. He said, I'd like to know more about your muscle force assessment in patients. So went out there and gave a talk. And there's Hank in one of the bear dens in Wyoming. They're actually in slash piles in rocks. So they are kind of like caves, which is different than Minnesota. So then went out there and, we started talking about hibernation in black bears. And they're really amazing, unique models because they don't lose any muscle mass, even they're immobilized for 4 to 6 weeks. And what I mean, if I opened up this table and that was a space underneath, that would be a big enough den for a 400-pound bear to be in there for 4 to 6 months. And we basically have collars with GPS on there, as well. We have implantable devices. We know how much they're moving around. They're immobilized. They're not leaving that as well. But what's interesting, they have an intact fight or flight response. So if you go arouse a black bear in Minnesota, they will leave the den, and they look just as fast in December as they do in March coming out of the den. So they don't lose any of this fight or flight abilities as well. So this is, University of Wyoming. We did necropsies on bears. I actually did a grizzly bear too. I only show this picture because I have a picture of myself dissecting a grizzly bear. No one else does probably in the room. And then we went to Bear Country, USA, and we did our pilot studies. And they were kind enough to anesthetize a bear. And again, we built a device. We didn't know how well it would work. So we did our pilot studies before we went out in the field, and tried to record this muscle force in hibernating bears in the wild. And so this is some of the dens we went to. So this is a rope we're supposed to hold on as we climb up to the den. And then they did use some pistols. The bears in Colorado, Wyoming, they can stand up and come out at you. And the only place they can come out is the entrance. So they would use pistols there to anesthetize the bear. So typically we'll use Telazol. But what was amazing is that we recorded this. We got funding from NSF and NASA. Again, that they really lost 25% of their muscle force. So these are your dorsiflexors. These are not anti-gravity muscles. These are postural muscles. And they've lost minimal force for this 3 to 4 month period of time that we studied them. Now, if you're the Department of Defense and you want to look at these parameters, they're going to take volunteers. You, and you are going to be immobilized for a month in a bed. We're going to give you food and drink, and we're going to track your muscle force loss. And you'll lose 0.7% per day. And so again, bears are not eating or drinking or defecating or urinating. So you can go about 3 days without drinking water. If you drink your urine, you can make it a week, right? Is that the one climber that, cut off his own arm, was trapped in a crevice in Colorado. And if you have a full drink, you can go about 30 to 40 days without eating before you'll die. And they're going, 4 to 6 months. So how we get the bears is we have barrel traps, and then the doors will slam down as they pull the food out of the back. You run your trap line, you drug them, and in the summer you use ketamine, and then you're going to put a collar on them. These are satellite collars, and then you can use Google Maps, and we can track where they are. We have about 40 bears collared in Minnesota right now, and about 25 of those have cardiac monitors implanted in them. Links. And yep, links as well. And then basically you can watch the bear recover. And I think Jenna was there for that study. So again, 4 to 6 months, no food, no water, don't urinate or defecate. Their core temperature remains near normal. So their core temperature goes from 37, 38 degrees centigrade down to 35 or 36 degrees centigrade. And so basically they can arouse and come out, and there's really no physical activity of them in the dens during that period of time. Amazing animals. This is the advantage of putting a collar on a bear and ear tags is there's about 15,000 bears in Minnesota. And this is the oldest documented bear anywhere in the world was in Minnesota. It was 39 and a half years old. And basically the DNR, this Dave Garcelis with her at age 37, tracked her for 32 years. They called her at age 7, took a tooth. They actually just aged the animal, like looking rings on a tree, rings on the tooth. And they were able to get a mortality signal. We were able to go to the den, find the skull, some of the vertebrae, bring it back to the U, scan it, and make computational models of it. So if this bear's 39 and a half years old, what do you think the average age of a bear is in Minnesota? 15, good guess. It's actually 5 or 6 years, because there's an active hunting season, and the DNR manages all this harvest. So we actually work with them to study their physiology, reproduction, viability as well. And there's a lot of other states that have black bears in them as well. So then this is a den in the winter, or this is a den in the winter. I mean, last winter we had no snow, really, when we went in the spring. The year before we had snow up to my hip. And so we had to have snowshoes on, and you're carrying all your equipment there, and you'll be walking around trying to find the opening, and it's just snow. And all of a sudden you're walking, all of a sudden you'll sink in, and that's probably the opening. And you'll take shovels and you'll start pulling the snow back, and then you might hear the animal, and then you're gonna drug the animal. But they have all different types of den. We've studied the different ecosystems from conifer forest to deciduous forests to this is the Great Plains in northern Minnesota. It's a lot of farming. They're actually corn-fed bears. Some of the male bears were like 600 pounds up there as well. And so, it's kind of fun to go out. If you go to the den and they're covered with snow or not, they're always facing out. So you can use an infrared camera to see where they are in there, or just look in there with a flashlight. And that's typically what they're doing. And basically they're always facing out. Why? Yeah, defend themselves against predators. And in Minnesota, the natural predators are wolves. Wolves don't hibernate. Colorado, Wyoming, it's wolves and cougars. So they actually have to be able to come out and defend themselves. So this is Dave drugging an animal. We always flinch. You flinch no matter what. All right, kind of fun to hear that. This is another way to drug the animal or surprise them. All right, we don't do it that way at all. This is another video where we got a laser on there. You put a cover over so you make it dark in there, so you can see the bear. Oh, jaw snapping. Scare us off. Hear a gurgly background. It's not a bad audio. That's the cubs in there. This is me going in there. Tim Lasky had a 360 camera. So this is me. You see the hip you're trying to get. Basically, you take a hypodermic with the Peelzall and you carve out a stick and you stick your arm in there, and then you're going to do a jab hole. And then you'll wait like 10 minutes. You're trying to always under drug the animal because they're mildly hypothermic, which also is anesthetic. And so then you go in there with a stick and poke them again to see if they're sleeping. And if they're not, then you got to re-drug them. But again, this was a video from a couple years ago when I drugged the animal. This is just another view of the whole thing. If you had a 360 camera, this would be the way to do it. We don't. So you actually are sticking your head in the hole as you're doing that to drug the animal. So again, they mate in May, June, July. There's delayed implantation of the fertilized egg until the end of November. 5-6 week gestation period. They're born in early January while the mother's hibernating. Wow. And she's in a deep hibernation because we've tracked all their behaviors with implantable devices. So this is Tim. We thought we would, try to design data loggers. This is before the Link project. We had defibrillator cans. We built the devices. They had in 2000 96 megabytes of memory, which was incredible. Right, this was state of the art in 2000. 96 megabytes of memory. And we actually had duty cycle these and do all the programming to figure out when they were going to turn on and everything else. But we got them implanted. However, we did anticipate that the bears would have a dramatic foreign body response and reject the devices in a couple-week period of time. So if you put that in a human and a calf and a pig and a sheep and a dog, the device will stay in until you pull it out. The bears had this massive foreign body response. And then we put these in December. And so the first year I put them up in the shoulder region with a lead down. I thought, okay, the typical implant zone. Didn't put the lead in the heart, just did externally. Didn't work. So next year I'm going to outsmart the bear. So I'll hang in the peritoneal space with the lead coming up. They marsupialized those cans and rejected them as well. And then there's yearlings in the dens, and they thought it was a fun chew toy. Shoot them all up. But we were able to get data from them, and bears have a dramatic respiratory sinus arrhythmia. So you all have one here. Their heart rate's 60 beats per minute. You inspire, it goes up to 61 or 62. Expire, it goes down to 60. A bear will have a resting heart rate of 5 or 10 beats per minute. When they take a breath in, the heart rate goes up to 60, 70 beats per minute. When they exhale, it'll pause. And that pause can be either 5 to 20 seconds long. We've seen some up to 35 seconds. And that's consistently through the winter for that particular animal. And so again, if you have a heartbeat for 5 or 6 seconds, you'll pass out. So these bears are more or less passing out after every breath they take. And their respiratory rate goes down to 2 or 3 breaths per minute. Huge conservation of energy. Again, they had this incredible healing ability during hibernation. These were some neck injuries that the animals have. We had some with gunshot wounds and arrows. If this was a collar you put on, and sometimes you size the collar for the animal to gain like typically 70, 80 pounds. This animal gained 150 pounds. And from April till October. Okay. So the collar injured them. I debrided it, I sewed it up, and in 3 months it's totally healed. Not only is it healed, but there's not just fibrotic inlay growth with scarring. They actually form new hair follicles around that area. And if you shave them, they don't regrow any of the hair. So they can put their energy only where they really need it. So if I shave a bear to do a 12 lead EKG, and we've done hundreds. If I do it in December, I don't have to reshave them again in March. And then we've done 12 lead EKGs. There's no electrical remodeling of the heart at all. I've done hundreds and echoes, if not thousands. There's no loss of cardiac mass, even though they're basically in a state of starvation and immobilized versus a patient, right? Patient ICU, you're going to lose 20, 30% of cardiac mass in a 3 to 4 week period of time. That's why you wanted your patients up in cardiac rehab as soon as possible. No remodeling at all of these bear hearts in the state of starvation. So then we went to the implantable reveal devices because we thought it was unethical to put the cans still in. And then we were able to record a lot more data. And so this is this pattern of intense respiratory sinus arrhythmia. Then we put on our GoPros, synchronized our watches, and we started walking towards the dens. And we could get 50 to 100 feet away, and they sense we're there. And we could be downwind, maybe we smelled bad from carrying all equipment or whatever, but they could sense we're there, and they start huffing, going, which drives up their heart rate, which then gets them ready for a fight or flight response. And then you give the drug, and then basically you knock out the autonomic nervous system. So then they have a constant denervated heart. The heart rates are somewhere between 90 and 100 beats per minute through the whole period of the anesthesia. So then you can record data for years. So this is just showing activity. So this is the summer active period. This is hibernation. This is on daytime, nighttime heart rates. Yeah, there's a funny spike right here. That's when we visited the den, which was really nice to know that the telazol lasted for 3 to 4 hours, wore off, and they go back right into that deep respiratory sinus arrhythmia. So like we were never there. So it's always nice to know as a field researcher, you have that. This is one animal we tracked for 6 years. This is a female bear. So she gave birth to cubs. And what they'll do is the cubs will stay with the mother as yearlings through the summer. Then they'll hibernate back with the mother. And then basically the following May, June, July, if she sees a good looking male, she chases off her yearlings and starts the process over again. So this last winter, we went to a whole bunch of yearling dens. One den had 5 yearlings. Their weights were, again, these were born in January. Now they're stopped feeding in October, somewhere between 70 and 100 pounds. And then a female that was 300 pounds. So we had 700 pounds of bear in this den that were drugging and pulling out. And this was one of the studies where Mark Dittmer did. We actually had the GPS satellite collars. We could take a drone, fly over the bear. The bear would look up at the camera and its heart rate would go up 2 or 3 fold. And the drone would leave and it would last for 10 minutes. So it's a huge catecholamine release. And basically, we were able to show that if we had a group of captive bears, same response, but they habituated after 2 or 3 drone flights. So again, the DNR is interested if they're going to track natural behavior of animals. You have to track that same animal multiple times because you are affecting the behavior by that drone flight. We can actually track road crossings, and they actually will stop at a road. The heart rates would go up. This one actually was hit by a car. We were able to get the device back. It showed by Gemini and then fibrillated, died from being hit by the car. And then now we're doing the Link devices, the Link 2 devices. And this is one of the things Tim and I figured out. We had these stations had bear dens where basically we can't put a device inside, a patient monitor inside the den, the bears will eat it. So you bury an antenna, it goes to a box sitting outside with a couple car batteries, patient monitor, and basically voltage regulators and timers hooked up to a solar panel to charge it and a cell phone module to send out the data. And this was the quality of data we were getting at the Medtronic BearLink site. And we've been able to do this for multiple years. And again, these pauses, just to think about it, they actually don't have any thrombotic events at all. And deep vein thrombosis or anything in the heart, even though the heart's static for that. And we've done ACT studies. They self-anticoagulate. The ACTs will be over 250 to 300. And we don't know if it's the fatty acids or what, or some of the proteins that are released. And that's a lot of group in Scandinavia are trying to study that as well. So again, huge fat deposits. They're only mildly hypothermic. They're still using tons of energy per day. There's protein turnover. And if you think of urea formation from the Krebs cycle, their kidneys are shut down. They have a specialized bacteria in the gut that catabolizes all the urea. Of course, NASA and everybody else is interested in it. There's groups, bear groups around the world studying all that. But again, they release these also hibernation induction triggers as well. And that's thought to be, opioid in nature. The active subunit is a delta opioid agonist. We've actually done studies on using that to protect skeletal muscle of the heart. And basically, it's thought to really be myocardial protective. They prevent prolonged organ preservation times, preserves muscle mass, and it minimizes both focal and global ischemia of the heart and other tissues. So again, we've been studying this for years in the lab and various preclinical protocols. This is Daniel Sig, who's now a distinguished scientist at Medtronic, but it's all these well-defined early and late phases of preconditioning. And this is through G protein cascades, affecting all the way down to potassium channel ATPases in the mitochondria and free radical scavenging. So there's a lot of basic work done on these mechanisms. And then they also upregulate the release of bile acids during hibernation. And that's been used in Eastern medicine for thousands of years for treating almost every disease, is bear bile. And actually there's black markets for bear gallbladders. You can sell them for $20,000 to $30,000. They actually do bear farming in Asia for this, but there are synthetic forms, tutka, that's used for treating liver disease in the United States. But basically, they will release more of that during hibernation. And again, so it's this organic mechanism. Here's just another bear. These are all asleep. So the cubs are kind of tucked under her. She's protecting them. She will not leave this den. She will protect her cubs. There's no snow on this one, but we've gone around to many dens where there's wolf prints all the way around circling the dens. And she's protecting her cubs down inside. So again, we can have blizzard conditions, and I'm doing these implants and everything else. We've had 20 below temperatures, 25, 30 below wind chills, snowing, and you're trying to do this field research at that period of time. So that's can be challenging. Some of the delta opioid research we've done, also the fatty acids, we've done infarct models. Daniel Sig and James Coles did that, where basically we would look at the area at risk. We would pre-treat these either IV or in the pericardial space, and we could reduce infarct size by over 50%, and arrhythmias during reperfusion by over 50%. So this is a ischemic reperfusion model that's been well defined in the literature. So this is just with these agents, which is really kind of exciting. And so you can do this actually in the pericardial space. We've done that with other antiarrhythmic agents, and it's a target site, and you can have the benefits of that agent in the target without its systemic effects. So we've been, again, then looking at putting bare bile acids in the pericardial space, fatty acids, delta opioids, or combinations. Today, or last week, we put in omegavan in the pericardial space for an hour prior to the isolation reanimation. It reduces arrhythmias, it reduces global ischemia, so the hearts will last longer once we have them on the apparatus, and it reduces the defib shock thresholds. So fewer shocks to get that heart back into native sinus rhythm, so minimizing myocardial stunning with fewer shocks. And then we're trying to apply this to all the things we're doing in the lab. This is another set of the isolated lungs, the organ care systems, where they're looking at that as post-conditioning agents there. We're doing that in the benchtop and the heart and the lungs. We've done post-conditioning in the lungs. So a human heart comes in now. It takes me about an hour to cannulate it, set it all up. And as we're warming it, we actually give the omegavan in the perfusate. And we can do high levels. You can't go too high in a patient IV because there's hemolytic properties of the fatty acids on red blood cells. So you minimize that, but a target organ or in a acellular perfusate, we can go ahead and do that. We've actually showed improved function. We've had hearts come in described to be an AF, and they actually come in. Some of them have actually gone into a native sinus rhythm for a while until we've washed out that the omega-3 fatty acids. So just kind of to summarize, our laboratory really focuses on translational research, cardiac anatomy, organ preservation, ischemic protection, and we've been studying hibernating black bears. They're these amazing animals and relative to hibernation. They don't lose any skeletal cardiac mass, state of total starvation, intact fight or flight responses, can come out and defend themselves at any time. And we're actually using this in the lab routinely. And so we reanimate a heart in the lab. It's not trivial. We're spending probably $10,000 to $15,000 a day. So anything we can do to optimize that is advantage for us. And we actually have a planned clinical trial where they're going to do CABG procedures, and they're going to actually do the pericardial delivery prior to doing the whole CABG to see if we can improve outcomes and time in the ICU of patients, and do that acutely in the lab. So last few fun videos here for you. Another bear in the spring. This is a bear that hasn't eaten or drank and defecated, urinated, or moved for 4 to 6 months. It looks pretty capable there. Actually fit the camera. Then we have den cams. And so these are a bear with yearlings. They're emerging then in the spring, and they're actually pulling dry grass back in because there's moisture in there as things melt out. My hypothesis is it takes the bears several weeks to leave, not because there's not enough food and energy out there. It's because they actually have to sleep. They're sleep deprived because they didn't have a natural REM sleep or any sleep for 4 to 6 months. And so we're trying to collect the data from our devices and everything else to be able to show this emergence from this deep respiratory sinus rhythmia takes several weeks as well. And we've collected the data and we're synchronizing it with our videos. We're collaborating with the University of Washington. We've got Link, the Link 2 device. This is Charlie Robbins. He's basically looking at oxygen consumption. They've got grizzly bears trained. They're feeding them honey through a tube, and the bear will then just walk on the treadmill. And we're trying to work with them to put the devices in to see if we could do this in the wild. So we're working with the wildlife groups at Yellowstone and Alaska Wildlife, trying to get these devices and collars so we can do field work. So we've actually made prototype devices there that have Bluetooth links to the devices implanted in animals. We can collect data for several weeks. We just don't have the battery capacity yet. We're working on that, but Tim Lasky's done this on elephants now in Thailand. They have plenty of room for batteries. They just having problems with the communications now, signals. So we're continuing to work on that. But that's maybe kind of the future direction as well. So again, now you're taking human clinical devices and you're applying it to wildlife research. And actually, Dr. Alina Katinsky, who's an electrophysiologist in the Chicago area, basically is doing that in the apes, great apes, and all the zoos across the US. Because they do show heart disease when they get into their 20s and 30s. So again, it's going the other direction now. And then this is just some more, this is my sterile technique for implanting the Link 2 devices. This is the data quality that we can get right in the field. And this last winter, I implanted 6, so I don't have a mouse.
Okay.
All right. Do you have the mouse back there? Anybody? Oh, sorry.
Yeah, we see it.
Pretty much
There you
done.
go, there you go.
And again,
Hit cancel.
Yeah, it's probably a big video for the.
all right.
Let's see.
So again, you also have to have fun and research. Every time we get a bear back in, we do the safety dance. And then basically, you know, some of the vehicles, this is how much snow. I have my snowshoes on, tinned in. So this is challenging. We take all our equipment out there. Our packs are weighing 50, 60 pounds. So you feel like you're in a Natural Geographic film every time you go out there, and it's super cool. But then to think about that of how you can bring it back to your preclinical research or human trials is, fun, things to think about and try to apply and get to. So thank you very much for your time.
Well, that was incredible. Thank you so much for sharing your insight. I think for many of us, we didn't realize how interesting black bears are and how much we can learn that we can apply to our patients. I don't know if there are any questions. Evan?
Thank you for an absolutely spectacular lecture. In your bears that are sleeping or hibernating with a marked sinus arrhythmia, do you see bradycardia-dependent arrhythmias, atrial fibrillation, anything of that sort?
Nothing, which is amazing. And then some of these animals are, again, that we are tracking with the ear tags and collars are 20, 25 years old. And they basically have no arrhythmias. They'll go from this pause right into a consistent native sinus arrhythmia. And we've really seen no electrical changes. There was a couple that might have a double peak, funny QRS complex, but it was a rare case. So it's mind-boggling, actually.
Do you have any clue to why the lifespan is so short? I know that in the wild, many of them are predated, but let's say kept in captivity, what is the natural cause of that? How long they live? Why?
Yeah, I have no idea. It's just supposed to be the natural course of their DNA and everything else. And, it's just like why do pigs only live 5 or 6 years, right? And it's just, I don't know, they're mostly healthy. We haven't seen any real disease per se, like any cancers or anything else. That sometimes they are covered with ticks, so they might have Lyme disease and things like that, but they don't seem to present with anything like that. So yeah, I have no clue. A lot to learn still. Yeah.
Do bears held in captivity, like in zoos, do they hibernate?
That's a great question. No. And actually, there's black bears in Florida, and black bears in Florida do not hibernate unless they're pregnant females. The pregnant females will go in and hibernate during that period of gestation and when the cubs are born. So there's 5, 6 weeks. She's kind of hibernating, and then basically while these cubs are nursing and everything else, she doesn't want to do any work. They said they're hibernating. I'm just anecdotal from what I've heard from the biologists down in Florida, but yeah, it depends on there. So the grizzly bears and the hibernaculums have access to water and food if they want, and so they don't really hibernate in these hibernaculums.
Yeah, absolutely inspirational. I'm almost speechless. 2 questions. Vascular aging, you don't see that in the bears. And the other question is, we use the transmatics here for transplant a lot. And this may be too clinical a question. I'm just curious. Maybe aware that there's more primary graft dysfunction after the transplant when you have used the device, which is probably what you're targeting with these agents. Do you have any hypotheses about why that might be and how that can be? How are you fixing that?
Yeah, well, I mean, there's a lot of research on looking at using different agents as well. But even they want to do some molecular kind of modulations of it. But again, this is, I think it's, you're putting packed red blood cells in most of these cases. And if you look at these packed red blood cells, they do degrade over time. So either they're gonna have to come up with a system of replenishing that or adding additional agents. But we actually did research on this organ care systems with whole blood. We were able to take the lungs out of a pig and keep them viable for 24 hours with whole blood. We actually took some of the lungs out of those animals, 5 animals, took one off the device 24 hours later, implanted another animal, and it was viable for 5-6 hours. We were able to get 10 sets of human lungs and 1.5 liters of blood from the upper body, because the transplant abdominal team didn't want us taking any blood from below.
Know that.
We were able to get 1.5 liters of blood and get that blood and the organ to the lab within 2 hours on ice. And we were able to keep viable function for 24 hours in these. So there's these other factors in the plasma and everything else that need to be investigated and looked at. And I think they're actually removing that, and they're actually maybe causing some graft dysfunction because they're removing too much out of their preparations. So our research hasn't gone anywhere. No one's adopting that because logistically it's hard to think about how you would do that, right? So you would actually have to have an organ that you put on there with the whole blood, and then that you're thinking you're going to go beyond. So the next step of that preclinical research is super tricky. How are you going to justify putting lungs on with whole blood for 24 hours and then transplanting them? So that's where we're the roadblock, right? So sometimes in research you hit these roadblocks in your clinical research.
Great. Mary, were there any questions online from anybody online? I don't know if anybody has access to the online thing. No, Mari's not here. Oh, well. No other questions? Paul, once again, incredible. I think we all learned so much. Thank you for taking the time to come to New York. And yeah, we look forward to having you here again in the future.
All right, perfect. Thank you.
Great to see you.
And once in a while, if you do something with blood, I would be interested. The disadvantage of having blood is you can see, but you cannot move up.
Very inspired by this. It was a great experience a year ago.
So we have to come back.
It's amazing. It's amazing.
Really appreciate it. Thank you so much. Yeah, I'm going to give this to Alex.
Yeah, take one.
All right.
All right. I know. All right.
I think we should arrange.
Yeah, it's all good. We're perfect.
I'll see you.
Thank you very much.
Yeah,
I'm gonna be there too.
okay.
But in that meeting, it's just a small room. So that's why I'm letting you talk to them.
Good.
Yeah.
Take care.
Thank you very interesting.
No clock in here, so I didn't have any contents here either.
Good to see you soon, maybe
For sure.
Work it out.
I did a great job.
That's
that's the whole Zoom thing, you know, there's always something.
Oh, come on, I'll push it. I'll push it.
Oh,
I'll
thank
push
you.
it.
Yes.
Put it on, I run the translate.
Okay. All right. Yeah.
I'm very inspired.
Good.
Thanks for coming.
You bet.
You want to vote? You got plenty of-
I just put my.
Yeah, thank you. Now, Mandy, you can go, Mandy. Mandy, you too, Mandy, what you need?
Let's go now.
Yeah, yeah, but did anybody follow those? Yes, please.
Water.
Okay, thank you so much.
That's it.